Assalamu alaikum everyone. My name is Muhammad Sarwar Tanveer. My topic of presentation is lipids, which is presenting to Dr. Shehbaz Ali. First of all, we talk about general introduction of lipids. Lipids are a heterogeneous group of organic compounds that are insoluble in water and soluble in non-polar organic solvents like benzene, ether, etc. Lipids are the one class of large biomolecules that does not consist of polymers. They have little or no affinity for water. The hydrophobic behavior of lipids is based on their molecular structure. Although they may have some polar bonds associated with oxygen, lipids consist mostly of hydrocarbons. Smaller than true macromolecules, lipids are highly varied in both form and functions. Lipids are include of waxes and other certain pigments, but we will focus on the most biological important types of lipids such as fats, phospholipids and steroids. Now we move towards the types of lipids. First of all is fats, second is phospholipids, third is glycolipids, fourth is steroids, fifth is waxes and sixth is triglycerides. First of all, we talk about fats. Fats are the esters of fatty acid with glycerol. It consists of many three fatty acid molecule and one glycerol which attach together and form a fat molecule. Although fats are not polymers, they are large molecules and they are assembled with smaller molecules by dehydration reaction. Unlike in carbohydrates or protein, there is other peptide bond interactions. In fats, there is a dehydration reaction. Dehydration reaction mean removal of water. When three fatty acid and one glycerol molecules attach, bond occur and a one water molecule removes. A fat is constructed from two kinds of smaller molecules, glycerol and fatty acid. Glycerol is an alcohol with 3 carbon, each bearing a hydroxyl group. Fatty acid is a long carbon chain which varies from 16 to 18 carbon atom in length. At the end of the fatty acid is carboxyl group, the functional group that gives these molecules the name fatty acid. Attached to the carboxyl group is a long hydrocarbon chain. The number of CH bonds in the hydrocarbon chains of a fatty acid are the reason of fats are hydrophobic. CH bond जो इन फैट्स के अंदर है उसकी वजह से जो है वो हाइड्रोफोबिक नेचर है फैट्स की. Fats separate from water because the water molecules have hydrogen bond to one another and exclude the fats in the reaction. Now we talk about the synthesis of fats. In making fat, three fatty acid molecules each join to glycerol by an ester linkage, a bond between a hydroxyl group and a carboxyl group. The resulting fat, also called a triglycerol, thus consists of these fatty acid linked to one glycerol molecule. The fatty acid is a fat can be same as in they can be of 2 or 3 different kinds. This is all about the synthesis of fats. Now the other slide is types of fatty acid. Fatty acids vary in length or in number and location of double bond. Saturated fats, unsaturated fat. Talk about the saturated fats, they have no double bond in their structure. Unsaturated fats have double bond in their structure. Classified on the basis of structure of the hydrocarbon chains of the fatty acids. If there are no double bond between certain carbon atom composing the chain, then as many hydrogen atoms as possible are bonded to the carbon skeleton. Such a structure is described as being saturated with hydrogen, so the resulting fatty acid is called saturated fatty acid. An unsaturated fatty acid has one or more double bond formed by the removal of hydrogen atom from the carbon skeleton. The fatty acid will have a link in its carbon atom chain where a cis double bond occurs. Now we talk about the types of fat. In the previous slide we discussed types of fatty acid according to the structure or depend on the position of double bond, either single or double bond present in the structure of fat. Types of fat animal fats or plant fats. These are the two kind of the on the basis of its physical properties. The in previous slide we discussed about the number of location of double bond or a single bond is kind varied in the form of these kinds. Now that other is on the basis of physical structure. Animal two types, animal and plant fats. Animal fats are saturated and solidify at room temperature like butter. Plant and fish fats are unsaturated and remain as a liquid at room temperature, e.g. olive oil or mustard oil. The kinks where the cis double bonds are located prevent the molecule from packing together closely enough to solidify at room temperature. Hydrogenated vegetable oils. Unsaturated fats synthetically converted to saturated fats by adding hydrogen. Peanut butter, margarine, and many other products are hydrogenated to prevent lipid from separating out in liquid form. Now the next slide is the function of fat. First of all, the function of fat is the major function of fat is energy storage. A gram of fat stores more than twice as much energy as a gram of polysaccharide such as starch. So that it is a most valuable for our body. Third is humans and other mammals stock their long-term food reserves in adipose cells which swell and shrink as a fat is deposited with and withdrawn from storage. Number 4 is in addition to storing energy, adipose tissue also cushions such vital organs as the kidney and a layer of its fat beneath the skin insulate the body. If we see any organ in animals or a human, we saw the layer of fat in a organ to prevent it from any rupture or any kind of spottage. The main energy sources like lipid store in this adipose tissue. Number 5 is the subcutaneous layer is especially thick in whales, seals and many other marine animals protecting them from cold ocean water. Now the next type of lipid is phospholipid. First of all, its general overview. Number 1 is a phospholipid is similar to a fat but has only two fatty acids attached to glycerol rather than three. If we talk about previously that fats are consist of three fatty acid and one glycerol. Unlike the fats, phospholipids have one glycerol and two fatty acid. Number second is the third hydroxyl group of glycerol is joined to a phosphate group which has a negative electric charge. Its valency is negative. Phosphate group PO4 negative. So number third is additional small molecule usually charged or polar can be linked to phosphate group to form a variety of phospholipids. Next is the general property of lipid. Hydrophobicity of lipids. A phospholipid show ambivalent behavior towards water, means strange behavior towards water. It has two parts, hydrophilic head and hydrophobic tail. Hydrophilic mean water loving part, hydrophobic mean water repellent या water hater whatever जो. Their hydrocarbon tails are hydrophobic and are excluded from water. Hydrophobic head is made up of phosphate group and its attachment have an affinity for water. Hydrophilic is made up of phosphate group which is water loving part. When phospholipids are added to water, they self assemble into double layered aggregates bilayer that shield their hydrophobic portion from water. Mean they form covering over them to prevent it water to form bond with them. Last part of the lipids is steroids. Steroids are lipids characterized by a carbon skeleton consisting of four fused rings. It's a basic structure physically जो है steroids का they join by the joining of four ring structures. Number second is different steroids vary in the functional group attached to this ensemble of a ring. Like if we talk about the type of steroid, which is cholesterol is a common component of animal cell membrane, precursor from which other steroids are synthesized. Number third is many hormones including vertebrate sex hormones like progesterone, testosterone are steroids produced from cholesterol. A high level of it is in the blood may contribute to atherosclerosis is a disease of vessels in which thickening of vessels occur due to deposition of cholesterol. Last is both saturated fats and trans fats exert their negative impact on health by affecting cholesterol level. This is all about the steroids.